Hello, my name is Heyu Lin. I am a PhD student at Melbourne University. My research interests focus on mercury methylation in microaerobic environments. Here, I'm going to briefly talk about my recent work on looking for new mercury methylators in low-oxygen marine environments. Let's get started. This project is to investigate the potential for methylmercury production by microorganisms in marine environments. First of all, I'd like to give you a general background. Mercury is a major pollutant for humans and the environment. It can occur naturally and can also occur from human activities. Methylmercury is a potent neurotoxin that bioaccumulates through food webs, which means humans could be easily exposed to methylmercury when we eat at the top of food chain. Mercury methylation was considered mainly conducted by anaerobic microorganisms in sediments, which required HECA and HECB genes. Ocean is the main source of global mercury. Methylmercury has been found to occur in suboxic waters, especially oxygen gradients at the upper and middle seawater layers. However, the methylators in that environment still remain unclear. A paper published before mentioned several HECA genes could be found in Saanich Inlet seawater, but the genes' hosts are unknown. Saanich Inlet is a seasonally anoxic field in Canada. We got large-scale multi-omic samples from Saanich Inlet, oxygen gradients spanning 6 major depths from the surface to 200 meters depth. We did binning according to differential coverages to recover metagenome assembled genomes, or MACs. We built protein homology models to predict the functionality of the novel HECA genes. We also measured the total mercury and methylmercury concentration profiles in Saanich Inlet water columns. Let's see the results. We finally got 56 MACs that carry HECA genes. These putative mercury methylators were from 7 clusters, in which the phyla Mercury-Microbia, Colitrichiota, and the SA324 clade have never been reported as methylators before. We found HECA-carrying Mercury-Microbia present in most samples and were the dominant mercury methylators. It occupied up to 2% of the whole community. Besides, mercury methylators were found more abundant and active in the benthic zone. This result is corresponding to our mercury analysis. As you can see, the Saanich Inlet contains zones of methylmercury production below 100 meters, where up to 17% of total mercury was methylated. Although the methylmercury concentration sharply decreased in the bottom water, which seemingly contrasted to our previous results. This may be explained by an increase of MERB genes that demethylate methylmercury to divalent mercury. By homology modeling, we found HECA could interact with cobalamin that enables transferring methyl groups. And HECB were found to bind with 4 iron 4 sulfur clusters, which could deliver electrons to HECA. Our novel HECA and B proteins are similar to the reported functional proteins, which strongly supports the functionality. From the tree of mercury-microbia genomes, we found all HECA-carrying mercury-microbia were clustered together to form a new clade. We further searched for our novel mercury-microbia clade in public databases. We surprisingly found it widely distributed globally in various environments. Finally, we found methylators contains multiple terminal oxidases in aerobic respiratory chains, enabling bacteria to survive under both aerobic and microaerobic conditions. The presence of other enzymes in these MACs suggests their key relationships to major biogeochemical cycles. To sum up, first, We provided more support for non-anaerobic microbial mercury methylation. Second, this is the first report that novel phyla marine microbia called Escherichia and SAR324 contain HGCAB genes and could play important roles in mercury cycling in marine environments. Third, modeling of the novel HGCAB protein structures supports functionality for mercury methylation. Fourth, marine microbia is the most abundant and active mercury methylator in salinage inlet oxygen gradients. It also widely distributed across different habitats. Finally, many thanks to my collaborators. This work cannot be done without their helps. That's all about this project. Thanks for listening, and I truly hope everyone stays safe and healthy in this special period. Thanks.